Okay, good evening, everyone. Good evening, good evening. So, if everyone quickly recall, friends, in the yesterday class, we have started studying about the enzymes that are involved in the process of the DNA replication. If all the students quickly recall, we have studied different type of enzymes. And I told to all of you that among all the enzymes, one of the most important enzyme that I want all of you to remember is DNA polymerase enzyme. Every one of you very quickly answer the question. In case of a eukaryotic organism, how many number of the DNA polymerases are present? Every one of you. Quickly. In case of a eukaryotic organism, how many number of the DNA polymerases are present? There are total 5 are present. Everyone quickly answer that. Among 5 DNA polymerases, which is the most important DNA polymerase in the eukaryotic organism, what was that? What is the name of the DNA polymerase? That is very important, we studied. So that is called polymerase delta. Whereas in case of a prokaryotic organisms, friends, polymerase 3 is the most important. Please don't get confused with that particular point. Okay. Now, later we all studied very clearly, friends. What we studied? Every enzyme cannot act like a DNA polymerase enzyme. I hope you all people remember what I have told to all of you. If an enzyme wants to act like a DNA polymerase, every student remember, what does it require, friends? Compulsory. Remember, every one of you, it has to show the characteristics of processivity, polymerization, and proofreading. Whenever it shows all these properties, then only we can say that that is the most important polymerase among all. And I hope you all people remember, among all these properties, which is the most important property, friends, the most important property is the processivity. Don't get confused with that. Okay, now all the students listen. In the yesterday class, I have also told to all of you very clearly that if the DNA polymerase enzyme wants to work, there is a requirement of the 2 magnesium ions which are associated with the 3 aspartate residues. Please don't get confused with that particular point. And another very important point all the students has to remember is that, remember my words, in case of RNA polymerase also structure is same, but in case of RNA polymerase, magnesium can be replaced by what, friends? Zinc at particular times or sometimes. And if every one of you quickly recall, I told to all of you that there are 3 DNA polymerases are present in the prokaryotes, but among all, the polymerase 3 is the most important DNA polymerase enzyme. And what is the reason why we are calling polymerase 3 is the major enzyme? In the last class also, I told to all of you, one of the major reason is that the polymerization rate. Because of the polymerization rate only, and processivity especially, it is said to be the most important polymerase. Again, I am reminding to all the students, remember this particular point without fail. Every single DNA polymerase generally will show 2 functions. What I'm trying to explain you, friends, polymerase 1, polymerase 2, polymerase 3. These 3 enzymes are basically going to perform 2 functions. What is that function, friends? 5' to 3' polymerization, which means, friends, synthesizing the daughter strand in 5' to 3' direction. And the number 2, every polymerase enzyme that plays a very crucial role in what process? In the 3' to 5' proofreading or the exonuclease activity. These 2 are the common functions. Pol 1 will do these 2, Pol 2 will these 2, Pol 3 also will do these 2 functions. But let's see the differences. You people no need to remember any kind of a subunits. Just remember that Pol 3 is the one having the highest Pol 3 is the one having the highest number of subunits. It is having the subunits more than 10. Please don't get confused with that particular point. Now all the students remember my words. Look into the point. Every polymerase enzyme will do the proofreading in the 3' to 5', no doubt. So polymerase 1 is doing the proofreading, polymerase 2 is also doing the proofreading, and at the same time, friends, polymerase 3 also doing the proofreading. Is it or not? Of course, yes. Now, the point I want all the students has to remember for the exam, polymerase 1 having a special function. What is that particular special function performed by the polymerase 1? It can do the proofreading in 5' to 3' direction also. Remember everyone, this is the only enzyme. This is the only enzyme that can do the proofreading in 5' to 3'. This proofreading cannot be done by polymerase 2, polymerase 3. This is one of the unique statement, and I will prove and we will discuss why polymerase 1 will do the extra function. And very important reason why do we call polymerase 3 is the most important enzyme among all. Every student just pay attention and try to understand my statements very correctly without confusion. Because friends, DNA polymerase 3 will add more number of nucleotide in a single second. All the students look into the picture very correctly without confusion. Polymerase 1, before it get dissociated, listen everyone, it will add 16 to 20 nucleotides in single second. And polymerase 2 will add 40 nucleotide in single second. But you see this, polymerase 3 can add up to 1,000 nucleotides very easily, friends. That's an important and unique statement. And the major reason, processability. I told you, right? Processability means ability, capability of an enzyme. So polymerase 1, before it get dissociated from the DNA, before it get fall down from the DNA, how many nucleotide it can synthesize? Up to 200 nucleotide. And what about the polymerase 2? Polymerase 2 can synthesize 1500 nucleotide. And what about the polymerase 3? Look into the picture. Polymerase 3, before it is falling down, it can synthesize up to 5 lakhs of nucleotides. Friends, please don't get confused with that particular point. Now let's talk about one by one polymerase, briefly and elaboratively. The first and very important statement all the students has to remember, DNA polymerase is what kind of a polymerase? Hope every one of you remember, it is a DNA dependent DNA polymerase. Because friends, entire information is coming from where? Entire information is exclusively coming from the DNA only. That's why it is called DNA dependent DNA polymerase. This is the one which is discovered in the year 1958 by Arthur Kornberg. I hope you all people remember, right? This is the straightforward question which was given in your JL question paper. Right. Now, all the scientists thought that, every student remember, all the scientists thought that there is only one DNA polymerase enzyme is present in the bacteria. But later, they have identified polymerase 2, polymerase 3. Now, the question is very clear. When they have identified polymerase 2 and polymerase 3, friends, whenever they are doing some experiments by using what E. coli, whenever they are doing the experiments with mutated E. coli, then they observed that yes, there is a presence of Pol2 also, presence of Pol3 also. Friends, now we have Pol4, Pol5, recently Pol6 also identified. But the question is very clear. Even though many polymerases are present, always polymerase 3 is the principal enzyme. And what's the common point you all people has to remember? Pol1, Pol2, Pol3. I told you, right? Same function. 5' to 3' polymerization, nothing but synthesis of a daughter strand, and 3' to 5' proofreading, which we also call it as exonuclease activity. Please don't get confused with that particular point. Now all the students try to understand what exactly I'm trying to explain you. Again, they'll give the question in the exam. Remember, always remember my statements correctly. DNA polymerase 1 having what functions? Tell me. 5' to 3' polymerization, 3' to 5' proofreading. Okay, you got that. These two are a very common function. And what is the special function? Special function is that it will do the proofreading or exonuclease activity in the 5' to 3' also. Apart from this, a very important point, DNA polymerase 1 is the enzyme which fill the gaps. I'll tell you in the upcoming slide. Suppose this is one parental strand, this is one parental strand. You remember, we add the primers, right? We add the primer and daughter DNA get synthesized. Friends, at last, we have to remove the primers compulsory. Correct na? We remove the primers. The moment we remove the primers, so who filled that gap? Again, that gap is also filled with the nucleotides by the DNA polymerase 1 only. Again, they can also ask the question in the exam, which of the following DNA polymerase enzyme? Answer, all the students, think and answer my question. Don't be hurry. Which of the following or which DNA polymerase enzyme do not have any role in the DNA repair mechanism? Every student answer. Which DNA polymerase enzyme do not have any direct or indirect role in the DNA repair mechanism? See, every one of you, everyone. No, Srinivas, Paul to have a minimal role. Forgot everyone. How come you people forgot? PART-3 is the enzyme. PART-3 is the enzyme which do not have any role in the DNA damage. The major enzyme which is involved in the DNA repair mechanisms is polymerase 1. Polymerase 2 also involved in the DNA repair mechanism, but very, less accurate, less efficient compared to the POL1. So, all the students, listen, I will summarize the functions of a polymerase 1. Polymerase 1 will do the polymerization in 5' to 3'. It will do the proofreading in 3' to 5', and it will also do the extra proofreading in 5' to 3', and it is involved in what we can say that replacing the nucleotides, or replacing the gap, or refilling the gap. That's a very important statement. And correct. And another very important statement is what? Very important statement is that this is the enzyme that plays a key role in the DNA repair. Okay, you got that point. Now, what's the interesting fact here? Now, the interesting fact we all people has to understand why polymerase 2, why polymerase 3 is not involved in the 5' to 3' proofreading. Why only polymerase 2 is involved? What is the reason? Now all the students understand. So scientists also did not understand why the DNA polymerase 1 is showing the extra proofreading activity. In order to prove that, all the students listen. Hans Kleno is the name of the scientist. So remember everyone, Hans Kleno is the name of the scientist. He has taken some chemicals like a sabitylicin chemical and a chemical like a trypsin. So all the students look into this. Suppose example, this is a polymerase 1 enzyme. Whenever Hans Kleno taken this particular chemical, you know what he has done? He cut the polymerase 1 into 2 fragments. One small fragment and one large fragment. Now they got an idea. So see, look into this. Every student look into the diagram. This is the structure of a DNA polymerase 1. So this polymerase 1 cut at 323 amino acid sequences, and remaining will be 604 amino acid sequences. Just now we are all studying that DNA polymerase 1 having a very special activity. What is that special activity? 5' to 3' exonuclease activity or the proofreading activity. So friends, that extra activity can be performed by this small fragment. Okay, and what about the large fragment? Large fragment will do remaining two function, 5' to 3' polymerization and 3' to 5' proofreading. You know, most of the students, what they do is, what they think is, small fragment is performing the extra function. So that's why it is called clinofragment. Actually, this is not the clinofragment. Large fragment is called as clinofragment, which is doing the function of polymerization in 5' to 3' direction, and which is doing the proofreading in 3' to 5' direction. Please don't get confused with that particular point. Now, I have already told you in the last class only, why do we require the proofreading? And again, friends, again, the question that you can expect in the exam, which of the DNA polymerase enzyme involved in the nick translation? What, friends? Which of the following enzyme is involved in the nick translation? That is also a DNA polymerase enzyme 1 only. First of all, what is a Nick translation? All the students listen. This is a double-stranded DNA. There are some enzymes. Let's take an example of an enzyme called as DNase 1. It is a degradative enzyme. You know, friends, what the DNase 1 does? DNase 1 created a single-stranded break. You can see, right? There is a break got created. Okay, now you listen. You all people commonly know that what is the polymerase 1 function? I have told you, friends, it is only remove the primer. It is only refill the gap. Correct, na? So now you listen. What is this particular polymerase 1 is doing? See this? Polymerase 1 will show the proofreading in 5' to 3'. And most importantly, it is also refilling that particular gap. Now you only look into the picture, friends. There is a gap, you can see, right? This is the gap. Now, friends, we have to remove this, we have to refill this gap. Now, for that reason, all the students listen. This red color you can see, right? Imagine that is the primer. So all the students listen my words. So DNA polymerase 1 removing this primer. Listen, everyone. DNA polymerase 1 removing this primer. The moment it remove the primer, the red color one, it has to refill with what? It has to refill with nucleotides, DNA nucleotides. Now you listen. It refilled with the DNA nucleotides. At the same time, it resealed that particular gap also. So what my statement here? It is removed the primer and refilled that particular gap. At the same time, it sealed that particular nick also. This mechanism only called as nick translation, which means, friends, it is doing the proofreading activity and it is doing the polymerization activity both the same time. That is what exactly we can called nick translation. This nick translation cannot be performed by the PART-2 or PART-3. Only PART-1 will show it. Always remember that point. Now, see, now when you look into the, see, you're not understanding, Aishwarya. See, I am not using the word sealing, right? I'm using the word nick translation. I'm using the word nick translation sealing. That nick can be, this gap can be replaced by this particular polymerase 1. At the end, ligase will come and seal that gap by the phosphodiester bond. That is at the end. Just listen, just listen. I am talking about this gap. This gap which is created by a certain degradative enzymes called as DNases, that gap can be sealed by this particular polymerase 1. When we remove the primer, at the end, ligase will come and do that. You will understand in the upcoming slide. Just look into the point. Now, another very important statement is that, anyway, I'm going little faster, friends, because you know the concept very well and we don't have a greater time to explain very crystal clearly. Of course, I'll do it. Now, you listen. All the students keep the point in your mind. DNA polymerase 2 also having the two functions, right? What are those two functions? 5' to 3' polymerization, 3' to 5' proofreading. Correct, na? Yes, that is correct. Now, then the point is that DNA polymerase 2 having the low processivity, and very important point all the students has to remember for the exam. I can say that DNA polymerase 2 act like a backup enzyme. Friends, what is the meaning of a backup enzyme? Which means, friends, when the pol1 is not functional, when the pol3 is not functional, then the pol2 come into the picture, because we should not stop the replication under any circumstances. So in order to continue the process of replication and in order to continue the process of the proofreading, the pol2 will comes into the picture. That's why we can call it backup enzyme. It is also participating in the DNA repairing, but not efficient. The only enzyme that plays a key role in the DNA repair is only pol1. Now, the king of all the enzymes, the most important enzyme among all, that is called DNA polymerase 3 enzyme. All the students now remember my statements very correctly. Why do we call DNA polymerase 3 is the most important enzyme. Remember all the students. You know very well DNA polymerase 3 enzyme called as most important enzyme because of the high processivity. Every student quickly answer my question. How the DNA polymerase 3 will get highest processivity? What is the name of the protein that is involved in the processivity of the polymerase 3? Consider this question only, you will get in the exam. Every one of you, what is the name of the protein that increase the processivity of the DNA polymerase 3? How come you people forgot? Not even 1%. How come? Very good, very good. Correct, Narayani. Beta clamp. Beta clamp is the name of the protein. Forgot everyone. Huh? Is the name of the protein that increase the processivity. Now let me explain to all of you how. And we all studied that. Remember, DNA polymerase 3 is the enzyme having a highest polymerization activity. And this is the unique enzyme for the elongation process. I told you, right? DNA polymerase 3 enzyme having how many subunits? It is having 10 subunits. All the students look into this beautiful picture. This is a very important statement. This is one parent strand. This is another parent strand. Both parent strands are separated and giving rise to the daughter strand. Now, what most of the students will think, in order to synthesize one daughter strand, we require one polymerase 3. In order to synthesize another daughter strand, we require another polymerase 3, which is absolutely wrong statement. The point is very clear that the two daughter strands can be synthesized by a single DNA polymerase 3. How is that possible? Because DNA polymerase 3 is a dimer. I will explain to all the students. Listen. DNA polymerase having a different subunits. How many are there? More than 10. Is it necessary to remember all these subunits for the exam? It is mandatory. You have to remember every single subunit for the exam. Everyone pay attention. Look into the point very correctly. This is called as a core enzyme. All the students follow me. Core enzyme consists of what subunits? Alpha subunit, epsilon subunit, theta subunit. But the point is, friends, how many core subunits are there? That's the point, right? There are two core subunits are there. You can see that point. Correct, na? So this one core subunit will hold one parent strand, one daughter strand. Another core subunit will hold another parent strand and another daughter strand. I will show you in the process. We are just reading only enzymes. Now, you only try to understand what is the function of a core enzyme. It contains alpha, it contains epsilon, it contains theta. Friends, very simple point. Alpha will do the polymerization, 5' to 3'. And what about the epsilon? Epsilon will do the proofreading, 3' to 5'. And what about the theta? Theta don't involve in the proofreading or don't involve in the polymerization. Theta will just help the epsilon. That's all. See what we have written? Theta stimulates the editing function of the epsilon. Always remember this particular statement. Now all the students look into the structure very correctly without confusion. Look into the point. Because the more depth you understand, more easily you can answer the question. Alpha, epsilon, theta, alpha, epsilon, theta. These are all called as what, friends? Core subunits. Now, every one of you remember the two core subunits associated with a tau dimer. Friends, I'll explain twice, just focus. It is called a tau dimer or a tau protein. You all people has to understand me correctly. Tau protein. What is the function of a tau protein? Tau protein is holding the two core enzymes and connecting the two core enzymes to the gamma complex. It is connecting to what, friends? Gamma complex. I hope every student remember, right? Gamma complex is also called as a clamp loader. Now, to this clamp loader, finally, what will be attached? beta clamp. Now you all people has to ask me, sir, what is this gamma complex or the clamp loader will do? Every student listen. This gamma complex will take this beta clamp protein, will take this and add it on the core enzyme. There are many beta clamps are there. So on the leading strand beta clamp, on the lagging strand also beta clamp is there. Whenever this clamp loader will take the beta clamp and add it to the core enzyme, then the core enzyme starts working super fast and it will start working accurately with the highest processability. So keep the point in your mind. Without beta clamp, it is impossible. None of the core enzyme never ever show the process of the processability. Don't get confused with that particular point. Again, all the students try to understand me what exactly I'm saying. Listen, this is called what enzyme? Core enzyme. Correct, na? So friends, whenever the core enzyme connected to the tau dimer, you know what do we call it as? We call it as polymerase 3 prime. What do we call? Whenever the core enzyme connected to the tau dimer, we call it as the polymerase 3 prime. Friends, whenever the tau dimer connected to the gamma complex, you know what do we call it as? We call the polymerase 3 star. Finally, when the gamma complex connected to the beta clamp, then only we call it as a polymerase 3 holo enzyme. What is the meaning of a holo enzyme? Holo means complete enzyme. When the enzyme becomes completed, enzyme becomes completed only whenever it is associated with the beta clamp. Don't get confused with that particular point. Now look into the same thing I have written here. Alpha, epsilon, theta, 3 together. What do we call it as? I'm explaining, Aishwarya, look into this. Alpha, epsilon, theta together call it as core enzyme. Only core is called as these 3 units. When the core enzyme connected to the tau dimer, it is called as a polymerase 3 prime. When this entire tau dimer connected to the gamma complex, this is called gamma complex. What we can call gamma complex? Clamp loader. So when the tau dimer connected to the gamma complex, we called as a polymerase 3 star. Then finally, when it is associated with the beta clamp, which is most important one. So all together, what do we call it as, friends? All together we can call it polymerase 3 holo enzyme. Holo means what? Complete enzyme. So the point is very clear. You can call a polymerase enzyme is a complete enzyme only when it is associated with beta clamp. Without beta clamp, it never become complete. That's a very important statement. Now every student has to look into this beautiful picture. Now, because friends, they will ask a direct question in the exam. Which of the following protein? I'll show you one wonderful animation after the process. You listen. They will ask a question. Which is the name of the protein that increases the processivity? That is called sliding clamp or beta clamp. Now you people look into this beautiful picture. Look into this. How does this particular beta clamp look like? It look like a C-shaped structure. You can see, right? The red color and yellow color. That is only beta clamp. In the white color, you can see, right? That is the DNA. Now, every student look into this beautiful picture. Try to understand what exactly I'm explaining to all of you. This is a beta clamp, one subunit. This is the another subunit of the beta clamp. Friends, where the DNA is present, look into that. DNA is present very deep inside. You can see that this is where the DNA is present. Now, because friends, beta clamp is a very big protein. You know, beta clamp having a external diameter 80 angstrom and it is having a internal diameter 35 angstrom. But every student commonly know that what is the diameter of the DNA molecule? Every one of you know, right? The diameter of the DNA molecule is just 20 angstrom. Now look into this. Look into this. Whenever the DNA is passing through the beta clamp, you can see, right, how much space is present here. Still lot and lot of space is present. Now the question is that what happens to that space? The interesting point is that that particular space can be filled with water. Now you can ask me, sir, how does the beta clamp associated with the DNA? Every student commonly know that DNA is a negatively charged. And what about the beta clamp? Beta clamp is a protein. Correct na? Now what you have to remember? Protein contain amino acids. Every student know very well. Now that particular positively charged amino acid. Look into this. Positively charged amino acids that are present in the beta clamp, they will be associated with the negatively charged DNA molecule. Don't get confused with that particular point. Positively charged beta clamp associated with the negatively charged DNA molecule. That is how both will be associated. Now listen, what I told to all of you, very important statement for the exam. They can give a direct statement in the exam. Beta clamp association and dissociation is energy independent process, which is absolutely wrong statement. Every student keep the point in your mind. If the beta clamp want to place on the DNA core enzyme, compulsory require ATP. And if beta clamp want to be dissociated, if the beta clamp wants to come down, fall down, again there is a requirement of ATP. That's why we have given a statement that its assembly or the removal requires the use of an energy dependent process by the clamp loader. Please don't get confused with that particular point. I want all of you to understand the point very correctly. Now, one other very, very important statement is that, look into this. Now you tell me, this is example beta clamp. Do you think beta clamp will directly go and associate with the core enzyme? That is not possible. Someone has to take this beta clamp to keep it on the core enzyme. Now, who is taking that? That is the one we can call the clamp loader. We can call the gamma complex. All the students, if you understand this diagram, automatically you will get it. Every student focus on the diagram. This is called core enzyme, alpha, epsilon, theta. If the core enzyme, every student follow me correctly. If the core enzyme want to be functional, what do they require? They require compulsory beta clamp. This is the beta clamp. Now you only look into the diagram. Do you think beta clamp would directly go and attaches to the core enzyme? No. In between the beta clamp and the core enzyme, what is present? Gamma complex is present. That gamma complex only we call it as what? Clamp loader. You can see the picture very clearly. Initially, beta clamp will come and attaches to what, friends? Gamma complex. It never attaches to the core enzyme. Now all the students pay attention, try to understand me what exactly I'm saying. Whenever the beta clamp attaches to the gamma complex, this entire complex will come and attaches to the DNA. Now you listen, look into the point. The moment it attaches to the DNA molecule, look into the point. ATP hydrolysis occur. You can see this. ATP hydrolysis occur. Whenever ATP hydrolysis occur, friends, this gamma complex will come out. The gamma complex is getting completely dissociated. The moment gamma complex get dissociated, look into the picture. Now you can see, right? Beta clamp directly associated with the core enzyme. Now the core enzyme becomes so powerful. Now the core enzyme can continue the DNA synthesis without stopping. Now imagine. Now entire DNA synthesis is over. Now what we have to do, tell me? You have to separate the beta clamp. You have to separate the core enzyme. How is that possible? Every student listen. Now the core enzyme reached at the end of the DNA. Now you look into this. Again, gamma complex associated with ATP molecule. And see here, it will come and join in between the core enzyme. In between the beta clamp and core enzyme. Whenever it comes in the middle, both get separated. You can see that. So what I'm trying to explain you, if the beta clamp want to associate it with the core enzyme, it requires gamma complex. Similarly, if the core enzyme want to separate from the beta clamp, again there is a requirement of gamma complex or clamp loader. So their association require ATP, their dissociation also require ATP compulsory. Always remember, if no beta clamp, the DNA replication will stop in the middle itself. I'm just telling you some examples. In the laboratory during the PhD experiments, what we do, sometimes we will give the drugs. We will give a drug. What that drug will do, that drug will go and block the beta clamp. When it blocks the beta clamp, even though DNA polymerase 3 enzyme is present, the replication never happens. Why? Because polymerase 3 enzyme want to be functional, they require beta clamp. But what I have done, some antibiotics or the drugs, they will block the beta clamp instead of blocking the enzyme. So that much important beta clamp is. No need to get confused with that point. So right now, whatever I have explained to all of you friends, just only one enzyme I have explained to you. That is DNA polymerase enzyme. That too only in the prokaryotes. Pol1, Pol2 and Pol3. Don't get confused with that. Now you listen. In case of eukaryotes, how many DNA polymerases are there? 5 DNA polymerases are there. Now, all the students think and answer my question. You all people given the answer, right? Polymerase 3 is the enzyme which never ever involved in the DNA repair. This is what you all people told me. Now, all the students answer. Simple question I am asking you. In case of a eukaryotes, which DNA polymerase do not have a direct or indirect role in the DNA repair mechanisms? I am asking a question, which DNA polymerase enzyme do not have direct or indirect role in the repair mechanism? All the students keep the point in your mind. That is the polymerase delta. Polymerase delta is the most important enzyme that do not have any role. Now, what's the ultimate function of the polymerase delta? It is the one which is involved in the replication process. All the students quickly answer my question. What is the name of the enzyme that is added the primer in the prokaryotes? So this is one parent strand. This is another parent strand. Here we are adding one primer. Here we are adding one primer. What is the name of that enzyme? Very good. You can call it DNA-G. You can also call it as what enzyme? RNA primase enzyme. In case of a prokaryotic organism, every student know it. But friends, in case of a eukaryotic organism, this is the important point. In case of eukaryotic organism, we don't require any primase enzyme. Now the question is why? Why don't we require any kind of a primase enzyme? Because we are having a versatile enzyme that is called DNA polymerase alpha. You know what it is going to do? Polymerase alpha itself add the primer. Listen my words. Polymerase alpha itself add the primer. Friends, the moment it add the primer, look into the point. The moment it added the primer, see this? It will only start elongating the DNA. But friends, it won't elongate the DNA continuously. Why? It can add the nucleotides up to 20 to 50. That's all. Now you have to ask me a question. We will read in the process elaboratively. Polymerase alpha only doing the primer, adding the primer. It is only doing the replication. Now the question is that what is the use of the DNA polymerase delta now? As I told you that DNA polymerase alpha, even though it started adding the nucleotides, it can't add more than 20 to 50. What is the reason? The reason is very clear. Because of its low processivity. Because of the low processivity, it can't add. So that is the reason after addition of the 15 nucleotide, the polymerase alpha can be replaced by the delta. And friends, the point is very clear. The polymerase delta will be continued till the end because it is having the highest processivity. I strongly believe every one of you remember this. See, just now we studied that if the polymerase 3 enzyme want to be functional, it requires beta clamp, which is very important. Now all the students answer. This is also asked a question. If the polymerase delta want to be active, which protein is required? So I mean to say, what is the name of the protein that increase the processivity of the polymerase delta? Every student has to remember this. It is called PCNA. I'll tell you the upcoming slide. Proliferating cell nuclear antigen. What is that? Proliferating cell nuclear antigen is the protein which will increase the processivity of the polymerase delta. I want you to remember my words. PCNA is the only enzyme, only protein. Replace the alpha with delta. That's also very important statement. Again, all the students has to remember for the exam. They can ask a direct question in the exam. In case of a prokaryotic organism, which is the polymerase that is involved in the DNA repair mechanism? Number 1, Pol 1. Similarly, friends, in case of a eukaryotes, which polymerase enzyme that plays a key role in the DNA repair mechanism? That is called Pol beta. What is that? That is called as the Pol beta is the enzyme which is involved in the repair as well as the recombination. Very important, friends. It won't involved in the replication. Now, the next point is that polymerase epsilon. Friends, till today, we have not understand the mechanism of the polymerase epsilon correctly. Some books and some research is suggesting that polymerase epsilon sometimes synthesize leading strand, sometimes synthesize lagging strand, but it won't synthesize both strands. That's why we can say that it is involved in the replication, but not an accurate enzyme. But still, the function of the polymerase epsilon yet to be understood. Still, the research is in progress. Now, the point is that whatever the polymerase beta is the enzyme that plays a key role in the process of repair and recombination. But the very unique enzyme is what? Pol delta only. And every student know very well, you studied in the intermediate only, mitochondria having a DNA. Chloroplast are also having their own DNA because they are semi-autonomous organelles or the self-replicating organelles. So the point is very clear. They require their own DNA polymerase enzyme for the process of DNA replication. So again, the question can be expected. Which of the following DNA polymerase is involved in the mitochondrial DNA replication, which is called polymerase gamma? Keep the point in your mind. Now all the students look into the picture correctly. Every one of you focus. Polymerase alpha is the, listen, it is the enzyme associated with initiation. What I told you, this is the two parent strand. What the polymerase alpha is doing? It is adding the primer. What is the length of the primer? 8 to 12 nucleotides. That's all. Now, after that, what polymerase alpha is doing? It is only adding the nucleotide. You can see that. It is adding the nucleotides up to 20 or 25 nucleotides it will add. Maximum 200. 200 it won't add actually. Hardly 20 to 50 it will add. So later, alpha can be replaced by what? The versatile enzyme delta. Why the polymerase alpha cannot continue? The reason is very simple, low processivity. So now the question is very clear. When I use the word low processivity, which means, friends, polymerase alpha is not associated with the PCNA enzyme. That's an important question that you will expect here. And they will also ask the question in the exam. Which of the following eukaryotic DNA polymerase enzyme do not have the proofreading activity, do not have the exonuclease activity? That is also alpha. That's why, friends, we can't call alpha is the most important enzyme. We never call it as. Every student remember 8 to 12 nucleotide RNA primers. It will add it. After that, it will add few nucleotides. It is immediately replaced by delta. Why? Because of low processivity and no proofreading. Simple point it is. Then what's the most important one? Beta. Beta don't have any role in the replication. It never participates only. It's extremely meant for DNA repair. Now, what is the most versatile protein, the most unique protein? That is called polymerase delta. Why the polymerase delta is the most unique protein, friends? The reason, every one of you listen, because it is having the proofreading activity, it is having the polymerization activity, and it is having highest processivity. Why it is having the highest processivity? What is the reason? Because it is associated with an important protein that is called as PCNA, proliferating cell nuclear antigen. So it will do the proofreading, do the polymerization associated with PCNA. So it is so powerful. Now look into the point. Look into the point. What is the speciality of a polymerase epsilon? The interesting point is that polymerase epsilon will do the proofreading, no doubt. Polymerase epsilon will do the polymerization, no doubt. And very interesting point is that polymerase epsilon will do the proofreading, even though there is absence of PCNA. But still it is very highly processive. Remember my words. It is highly processive even in the absence of PCNA. Now the question is very clear. Sir, it is very highly processive even in the absence of PCNA. Then why don't we use this? Then why don't we use this? The reason is that sometimes it synthesizes only lagging strand, sometimes it synthesizes only leading strand. So that's why it can act as a substitute enzyme for the delta. That's why I can't call this as the major enzyme. It is always a substitute enzyme, even though it is a highly processive. Always remember that. Now every student listen my words. What is the function of PCNA? Whatever the function of the sliding clamp that will show in the E. coli, the same function performed by the PCNA. Simple point. Required for high processivity of polymerase. Now you remember, how does the beta sliding clamp added to the core enzyme? We all studied, right? By the help of clamp loader in case of prokaryotes. We can call it gamma complex. Now you tell me, what is the name of the clamp loader or the gamma complex in eukaryotes? We call it as RFC, replication factor C. Never ever get confused with that particular point. Then the next one, very important one, friends. What is the name of the protein that is involved in stabilizing the double-stranded, two single strands? We studied, right? SSBP, single-stranded binding proteins. What the single-stranding binding proteins will do? They will bind with the single-stranded DNA. They never allow the two strands to join. Friends, that SSBP having a separate name in the eukaryotes. That is, we call it as RPA, replication protein A. Always remember that particular point. Okay, now the question is very, very clear. No, Sanvika, that is wrong. That is whatever you said is right. We can say that the preventing coiling, that is true. Let me explain you in the upcoming slide in a little elaborative way. But if they ask the question preventing the torsion or preventing the coiling, we should choose the word topoisomerase. But if they ask a question, which one will remove the hairpin loop-like structure, hairpin, then we have to choose the SSBP. Let me explain in the upcoming slides. Now, every student remember, these are all the different names for the prokaryotic E. coli. I hope every student remember, right? This is the question which was asked in the your DL question paper. DNA G protein, which is the one which synthesizes the RNA primer. Friends, every student listen my statements. No need to confuse. Whenever I am explaining you the process, it will become very easy to all of you. You know that DNA replication, whether in a whether it is a prokaryotic organism or the eukaryotic organism, DNA replication will start from a particular point we can call it ORC. Now the question is that who will recognize this ORC? Who will recognize this ORC? The ORC can be recognized by DNA A protein. Sometimes this DNA A protein, you also call it as initiator protein. Never get confused. Next one, DNA B protein. DNA B protein is also called as what protein? Helicase. Every student remember my words. In case of prokaryotes, helicase is called as DNA B. In case of eukaryotes, helicase is called MCM protein. It is mandatory, you have to remember, friends. And the point is that DNA B is the ATP dependent enzyme. Who is guiding the DNA B? Every student remember, DNA B is guided by the DNA C. So friends, now point is very clear. If the DNA B protein, that is called helicase enzyme, if this enzyme want to be active, there is a requirement of DNA A protein also. There is a requirement of DNA C protein also. When these three are present, then only opening occur. And DNA G is the one which synthesizes the primer. SSB, in case of eukaryotes, what do we call RPA, which will bind to the single stranded. The versatile enzyme called Topo isomerase, which will prevent the supercoiling or remove the supercoiling. So far, friends, we have studied only one enzyme, that is polymerase enzyme in prokaryotes also, eukaryotes also. Now we have to study all other enzymes also. Whatever the information I am saying, all the students try to understand me correctly. Primase is the enzyme also called as DNAG in case of E. coli. In case of eukaryotes, just now I told you, right? DNA polymerase alpha having a primase activity. Simple point. What's the function of primase? You also know very well. The ultimate function of the primase is adding the primer. So friends, what is the length of this primer? The length of primer in case of a in vivo, I mean to say within the within the body, it is 11 plus or minus 1. But friends, in case of a in vitro, Sanvika very specific is 11 plus or minus 1 in the body. And if you look into the in vitro within the laboratory, especially we used in the PCR technique, right? There it will be up to 60 nucleotides. Now the primase enzyme will come and add the primer. Then the versatile DNA polymerase enzyme will come and synthesize the new strand. No need to get confused with that particular point. Now, friends, look into this. Next one is called helicase. In case of a prokaryotes, we call DNA B. In case of eukaryotes, we can call it MCM. Every student keep the point in your mind. You know, friends, helicase is a hexameric protein. Means it is having 6 subunits, 3, 4, 5, 6. It's like a wheel. Look into this. It is like a wheel. It's like a wheel. It's keep on moving very fast. You know how fast it will move? In 1 single second, just 1 second, it will break 1,000 base pairs. So it will break 1,000 base pairs in 1 second by like a wheel. It's keep on moving. Now the question is crystal clear. If it wants to move very fast, it requires energy compulsory. So that's why it is ATP dependent. Now friends, this DNA B or helicase continuously guided and monitored by the DNA C. Always remember that point. And from where the DNA B will start opening the double-stranded DNA? From the origin. Who will recognize the origin? Undoubtedly DNA A. So my point is that if the DNA B or the helicase want to be work, compulsory require DNA A and DNA C compulsory. Now all of you try to understand. Now Sanvika, look into this. What is the function of SSP? Single stranded protein. Every student listen my words, carefully understand what exactly I'm saying. The helicase enzyme open the 2 strands of the DNA. Okay, you got that. Now this is a single strand. Now this is a single strand. There is a possibility inside the cytoplasm, inside the nucleus, there are a lot of degradative enzymes are present. lot of degradative enzymes are present. You know what they will do? They will mostly try to digest the single strands. So in order to prevent the digestion, in order to prevent the degradation of the single strands, we will add this SSB. We will add this SSB, not only preventing from the enzymes. Again, there is a possibility, right? Two strands will rejoin again, right? So remember everyone, to prevent the double-stranded DNA formation, we don't allow the double-stranded to form again. So to prevent the double-stranded DNA formation and to prevent the single strands from the degradation, there is another possibility is also there. What was that? Now, when the two single strands are separated, there is a possibility that, friends, this single strand can form coiling structures like this, like this. You can see, right? Hairpin loop-like structures. So to remove these hairpin loop-like structures, to prevent the degradation from enzymes, to prevent the double-stranded DNA formation, we will add single-stranded binding proteins. And friends, how many single-stranded binding proteins we will add? If we add 1 or 2 proteins, do you think it is sufficient? No. We have to add nearly 60 SSB-BP proteins. And very interesting point is that all the single-stranded binding proteins will work in a cooperative fashion. You know, what do you mean by the word cooperative fashion? One protein is going to support the other protein. Now you look into this. One protein is added here. Okay, one protein is added. Now friends, this protein will provide the binding site for second protein. Now second protein will come and attaches. Now second protein will provide the binding site for third protein. Third protein will provide the binding site for the fourth protein, and so on. So that's why we can say that they are all working in a cooperative fashion. Don't get confused with that. Now the most unique enzyme. From this enzyme also, friends, there is a possibility that you can get very different type of questions. Listen, every students, just now I told you very clearly, right? Friends, helicase enzyme is opening the double-stranded DNA 1000 base pair per second. So fast it is moving. So it creates a lot of tension. Every student, imagine this diagram. On one side, you are holding the thread. On another side, another person is holding the thread. So by using the force, by using your thumb force, you are opening the thread. So you listen, you're creating a lot of pressure on the thread. So thread is opening continuously. At one particular point, you tell me, look into this, the thread undergone the coiling. Now, whenever you reach till this point, do you think you can open further? It is not possible, right? You can't open further. You have to cut it and you have to open it. Then only you can move further. Same thing you people imagine. In the place of the thread, you remember DNA. In the place of a hand, you remember helicase. So whenever the helicase enzyme is opening so fast, at one particular point, DNA undergo the coiling. You know, in this particular picture, the coiling is shown very lightly. You might observe, right? In the 5-6 years back, we are having a land phones. In the land phones, you might observe land phone having a wire, right? If you try to coil that wire, it can easily coiled very tightly. The DNA also undergo the coiling exactly in this way only. So many coilings will occur because of the helicase tension. Then this can be cut by a versatile enzyme, that is the topoisomerase enzyme. So this topoisomerase enzyme, in case of a prokaryotic organisms, we call gyrase. In case of prokaryotes, you call gyrase, but in case of a eukaryotic organism, you call it as the topoisomerase only. There are total 2 topoisomerases are there. Whatever the information I am telling you, friends, remember as it is. Okay, polymerase 1 and polymerase 2. I am not explaining till the net level. If you look into the CSIR net level, they will raise the questions only from the Topoisomerase mechanisms only. For JLDL set, they won't ask that, but listen. Polymerase 1 and 2, sorry, topoisomerase 1 and 2. Topoisomerase 1 further divided into 1A and 1B. Topoisomerase 2, it is only a single unit. Remember, topoisomerase 1 only cut a single strand, whereas topoisomerase 2 will cut a double strand. Remember, all the students, look into the picture correctly. No need to get confused with this point. This is the topoisomerase enzyme. Friends, again, they will ask the question in the exam. Which of the following amino acid present in the active structure of a topoisomerase? What is the question? Which of the following enzyme is present in the active site of the topoisomerase, which is the tyrosine? Every student look into the picture correctly. This is the DNA. Now you see this. Tyrosine having OH group, right? Every student know, right? Oxygen having a lone pair of electron. It is acting as what? Nucleophile. Now that nucleophile, look into this. It will come and cut the single strand. You can see that. The moment it cut the single strand, which strand got cut? Purple color strand got cut. Through the gap, the green color strand is moving. See the arrow, see the red color, whatever I'm drawing. Through that gap, the green color strand will move. You can see that the coiling got removed. Now you can ask me, sir, who will seal that? Undoubtedly, friends, ligase will come. Ligase will come and seal that. This is type 1A will do. What the type 1B will do, you know? See, every student listen. Type 1B will do the twisting. What it will do? Twisting. Every student listen. This is the type 1B. Look into this. First, it cut the single strand. Now listen, you can see, right? It cut the single strand. There is a break. So now you see, friends. See, every student remember my words. Originally, this is how the DNA should look like. Because of the coiling, DNA became like this, opposite. Remember, this is the orientation of the DNA originally. Because of the coiling, it became opposite. Now, what topoisomerase is doing, it is twisting back to this. That is what it is doing. See this. The two ends of the DNA double helix now rotate relative to each other. Don't get confused. Now the two strands of the DNA or two ends of the DNA double helix now rotate very relative to each other. As a result, friends, from here it will come back to the normal conformation. Then topoisomerase 1B will be released out. No need to confuse. It's very easy. Topoisomerase 1A will cut a single strand. Through that gap, it will pass the other strand. Ligase will come and seal the nick. We got that. I'll show you the animation. But 1B, once it create the break, it is twisting. It is twisting in such a way that it is rotating in such a way that whatever the direction it is there, it will twist to its original direction. As a result, what happened? The coiling will be removed out easily. Then the topoisomerase 1B will come out. Then what about the topoisomerase 2? They can also ask the question in the exam. Which of the following topoisomerase 2, which of the following topoisomerase will cut a double strand? This is the only topoisomerase. They will also ask the question, which of the following topoisomerase is ATP dependent? Every student remember 1A and 1B, they are not ATP dependent. But friends, topoisomerase 2 is the one which is ATP dependent. And very interesting point, topoisomerase 1A and 1B compulsory require the ligase. And the speciality of the topoisomerase 2 is that it do not require ligase. It is having intrinsic ligase activity. Remember, it cut the phosphodiester bonds on both strands of double-stranded DNA and release the supercoil. So by consumption of ATP, every student look into this picture. You all know very well, friends. DNA is negatively supercoiled. Correct na? See, friends, I'm telling you, DNA is a right-handed orientation like this. You can see, right? This is the right-handed orientation. But see, friends, because of the coiling, see this, now the orientation changed. Changed to which direction? Left hand. It is called DNA containing a positive supercoil. Now I have to convert the positive supercoil into negative supercoil. How will I do that? By an enzyme topoisomerase 2, also called as gyrase, having 2 units of gyrase A and 2 units of gyrase B. Now you look into the picture correctly. Now the 2 units of gyrase A and gyrase B attaches to that coil. Attaches to that coiling. Look into this. Now it created. Now it created a nick. It cut it. It cut the double strand. Friends, now you look into this beautiful picture. Look into that. Now what this subunit is doing, tell me. One A unit and one B unit. Now you see what it is doing. It is pulling the strand back of that other strand. You can see that. What this AB is doing. See this. It is pulling the strand back of the other strand. You can see that. As a result, friends, the coiling can be easily removed out. It's like a rubber band, what we do. Now, once it remove the coiling, topoisomerase 2 only will seal it. It do not require external ligase. It will only seal it on its own. No need to get confused. It's very simple. First, one A unit and one B unit, after cleaving, it will hold the strand and it pull it back. It pull it from the back side. As a result, coiling will be removed. The moment coiling will be removed, itself will seal it because it is having intrinsic ligase activity. The last and final enzyme, very important enzyme, ligase. Most of the students will think that ligase is the enzyme only involved in the replication. What is the question that is asked in the previous set exam? The question asked is, in which of the following steps ligase is not involved? So every student remember, ligase is involved in replication, ligase is involved in recombination, ligase is involved in the repair, ligase is involved in the splicing. In multiple steps, ligase is involved. See, ultimate goal is what, friends? Ultimate goal is formation of the phosphodiester bond. How do we form the phosphodiester bond? By joining the 3' hydroxyl group to the 5' phosphate group. So that is the common point. Again, friends, remember, do you think ligase enzyme can do the function very easily? No. Ligase enzyme also require energy. Either it will take the energy from NAD, or it will take from AMP, or it will take from the ATP. Mostly ligase also consume the energy from the ATP only. Please don't get confused. I have told you one statement, you remember. Topoisomerase enzyme, in its active site, it contains tyrosine. Correct, na? Similarly, friends, all the students remember, this ligase enzyme, in its active site, it contains an amino acid lysine. I want you to remember this compulsory. In its active site, it contains lysine. Please don't get confused with that. Okay. Every one of you quickly answer these questions very quickly. The principal polymerase needed for the DNA replication is? The principal polymerase involved in the DNA replication is? Friends, there is no doubt. I have to go little faster. Okay, I have to go little faster, then only we can do it. Okay, so everyone know, right? Okay, that's good. It is the polymerase 3. Okay, DNA unwinding is done by every one of you. DNA unwinding is done by. So everyone know that they're asking the unwinding. Unwinding can be done by helicase. Okay, now which enzymes remove the supercoiling in replicating DNA ahead of the replication fork? We can call Narayani, but mostly we can call gyrase only. You can call topoisomerase in prokaryotes also, but widely called gyrase. Okay, question number 4. Proofreading activity of the DNA polymerase 3 occurs via. Proofreading activity of the DNA polymerase 3 occurs via. So obviously it never be endonuclease. It is always exonuclease. 3' to 5' exonuclease activity or the proofreading activity. Okay. The reaction in DNA replication catalyzed by the DNA ligase is. So we all people know, right? DNA ligase is the enzyme. Always make the phosphodiester bond between 3' hydroxyl group to the 5' phosphate. Okay. Now all the students listen. By tomorrow class, I will finish the prokaryotic replication process and eukaryotic replication process. Now I will give you the very tough questions at the end of tomorrow class. At the end of tomorrow class, I'll give you very tough questions from both prokaryotic and eukaryotic replication. If you people can answer those questions, trust me, you can answer any question in the exam. Okay. So be prepared for that. Okay, I hope it's clear for everyone. Some people who are very new, they may feel very confused about this unit. I want you people to re-revise compulsory. Okay, okay, that's all for now. Yeah, I'll send it. Okay, I'll see you in the tomorrow morning 4:30 a.m. sharp. Okay, okay, thank you all of you. Thank you.